Okay.
Yeah, I already got the details.
So good morning, Carlos. We are going to start our the first presentation, and the aim of this presentation. So we want to encourage our young people for the for high impact journal. For this reason, actually, I give the title like this, like how we can get or how we can publish like a big journal, like a Nature review material. So the we can get some opportunity with the Carlos, and if he can share some of his hard working, like how he did and how he get the chance to publish there. Every beautiful work behind this, actually there are many dirty work, like we can only see the beauty of the paper, but behind this, behind the screen, there are many dirty work, like a lot of effort and also like a lot of hard work. So from this time, actually we would like to get this opportunity, like how Carlos how he get the chance to publish there. And if you guys have any question, it's please feel free to ask Carlos so many questions so that we can learn all the people. So little bit about the Carlos. Carlos Guimaris, and the last from his city, and he's a bioengineer. He done his master degree in molecular biology from Faculty of Engineering, University of Porto. Currently, he is a visiting scholar at Stanford University, and he is a PhD student. Within this time, he published like some good paper, and his research interest with biomaterial and tissue engineering. He's particularly focused on cell material interaction, microfluidic techniques for biofabrication, and high throughput scanning platform. He, as well as he also work with the hydrogel for biomedical application. I think currently we have some work at advanced material, which is under review now. And he's also some good publication. I think also Science Advance is under review. And he's planning to submit one more paper, like I think Nature Biotech. So he has many publication, and one of the like the good publication that I actually we want to discuss here, Nature review material. And this presentation will cover like how we can publish high impact journal. So enjoy, Carlos. Maybe you can continue. Please continue.
Thank you. Thank you
And
very much for the.
again, we would like to thank the Carlos because it's actually very early time. So
don't worry, I am the one. I am the one that I have to thank. I want to thank and
yes,
start exactly
Carlos.
there by acknowledging Rajiv for the invitation to be here. It's really a pleasure. I've never been, like I was saying, in Bangladesh. So hopefully one day, once all this coronavirus pandemic is gone, I can go there for a visit. I'll be very happy to visit the country. And so it's a pleasure to be able to talk to you today and to have you there. I know it's already quite late, so thank you for being here. And hopefully I will be able to show you not just the paper itself and how it is and how it looks, but like Rajiv was saying, the dirty work and the dirty job behind it and getting it done. Okay, so basically, we're going to be talking about this paper of ours, Stiffness of Living Tissues and its Implications for Tissue Engineering. And this is going to be where the talk is going to revolve about, okay? But I'm not just going to be showing portions of the paper, that would be boring, but try to gave you an overview of the editorial processes behind it, of the steps that go in publishing a review of this size, in terms of impact, not really document size, because you will see it was rather compressed, this document, but rather on the process behind it. And then I will go throughout the review in the topics we talked about, the scientific discussion that was there, how we outline different ideas. So hopefully you can also have a general view on how to construct a document like this in terms of information, where to put it, how to take advantage of tables and figures and boxes and so on. And maybe by the end, you can have a good perspective into something like this. So I'd like to start exactly by the process. So the process behind a document like this. And the steps behind this review, I'm going to show were the ones I dealt with personally. But basically, there are two ways that you can get a review with such high impact, okay? One is what I call the easy way. And the easy way is that you are invited to publish a review with them, okay? So they can go usually to professors they think are working on a certain field that they are interested in cover, and they will ask you and invite you to write a review. That's the easy way, because you'll see that brings you forth a lot of steps and closer to acceptance. But these are very high impact factor journals, and they are not inviting a lot of people. They invite very, very select people. So certainly they would not invite me, at least so far, and therefore the process was a little bit different and took advantage of the ability to do a proposal. All right. So if you're not at an invitation, you shouldn't write a paper and send a full paper. You should rather propose the idea of the paper and then see how that pans out. So that's exactly how it started. So we created a proposal, which was basically a review on the mechanics of different biological tissues. And this review was on the back of my head for a long time, because I was working already for quite some time with biomaterials and cells, mechanical properties, and I was always thinking and trying to find out whether certain materials had mechanics proximal or similar to certain tissues. And every time I wanted to see mechanical properties of skin or cartilage or bone, I'd have to scour through a lot of papers. Some would report things in a little bit erroneous way. Some would give me nano details, but I wanted micro or macro. So it was always very difficult to see the mechanics of a tissue quickly in order to assess and to compare to our material. So I always had this idea, why didn't anyone yet review all of this, and like all the living tissues and their mechanics in a single document? Okay. So that was basically the concept of this proposal. Then we also discussed and talked about going through different dimensions of analysis, different techniques, and a discussion, since we are tissue engineers, on how can we engineer these tissues based on this knowledge. Okay. So we started with this. This was our proposal. We submitted it to Nature Reviews Materials. And then this proposal goes through several stages. The first is basically passes through an editor that takes it up to an editorial board review. Okay. This editorial board review basically is a meeting of the editors, and they go through all proposals. They see several proposals, and eventually they select the ones they are interested in seeing a full manuscript. Okay, so if you're lucky, and this is the most critical step, they will decide on the proposal. And at this stage, if it is accepted, it's similar to an invitation, because they show interest in seeing the full manuscript, and you already know that the journal wants or might want to have a review on the topic you propose. So this is the most critical step, proposal. And the proposal should be, for a journal like this, it has to be extremely novel, okay? You shouldn't just bring along another review on a topic or just to gather a perspective on recently published papers. It should be something that has never been reviewed before, because that's what this kind of journal looks for. And of course, it's very hard to find. I have written other reviews, and of course, not all of them will end up in a journal like that, because to find a topic that has never been reviewed so far, it's something that it's not easy. But if you come across it, then you have a chance of doing something like that. So you should always pay attention. And like I said, if you feel there's something lacking in the field, a review that doesn't exist, that's a great opportunity to target a publication like that. Okay, so they decide on our proposal, and if it is positive, then they not only say yes or no, but they are actually going to give you suggestions of how to improve or how to make the manuscript interesting for them. Okay, so you remember all that I said that we wanted to review? They basically said, that's interesting. We want to look at that, but also integrate a perspective going from the single cell to the full organs. So discuss cell mechanics, extracellular matrix mechanics, cell mechanics, cell ECM mechanics, tissues, organs. and also discuss what happens in disease versus healthy tissues, okay, together with everything that you already proposed. Do it in 6,000 words with a maximum of 6 to 7 display items. Okay, and this was already a challenge because we had an initial document with the topics we proposed, and we were already over 10,000 words. They asked us to add 2 very big sections and make it 6,000. So at this point, we realized that we had to engineer the paper in a completely different way. We could not outline and describe this group did this group did that, because that would be impossible to fit there. So we had to shift the manuscript building around the display items, concentrate in figures and tables, all the main batches of information, and then use the few words we had to have the most meaningful discussion, to bring the most important points and comparisons, and so be able to fit everything we had in mind in there. Okay, so we did our paper, we wrote our paper, and from the outline, they gave us 2 to 3 months to have a full manuscript. So it was actually a relatively short time frame. And after having this first manuscript, we submit it, and then it goes to peer review. In a journal like this, it's always going to be something like 4 or more reviewers. So in this case, we had 4 peer reviewers, and they really scoured the paper from top to bottom. It's not just like general comments and small points. They actually allowed and gave interesting feedback, which allowed us to do a revised manuscript version and integrate all of these different perspectives. They asked us to add topics that were missing, that they said was mandatory to have in a review like that. So the peer reviewers actually had a hard work, and they were contributing to make it a better paper. So that was something that you don't see always. Okay, we revised the manuscript, and they said focus mostly on the parts of cell mechanics and ECI mechanics, and we gave a second revised manuscript, which then went to peer review again. They still made some additions. We had to revise the manuscript once again, submit this version, and then in Nature there's something very particular, which is they take your paper to designers, and they're going to go through all your figures, and they're going to design it in a new way that looks better and fits the journal. And then hopefully they send the figures to you. They send manuscript proofs, which can be heavily edited depending on the state of the manuscript. Editors are gonna rearrange your sentences. They're maybe moving sections around. They really work on the paper. Everyone has a big effort to make this an interesting publication for a big audience. And if you validate their proofs, you accept, you like the figures, eventually you reach publication. Okay, so this is a general perspective of Nature reviews. And this whole time frame was of about 1 year between proposal and publication. So it took some time, even though we wrote the manuscript in 2-3 months by their decision. All these peer review stages, they can always take like a month. So the process can be long enough. But the manuscript in the end, the proposal, you could say it was like a little peach, and then the publication was a big watermelon, you know, so it really grew a lot. It was a big fish. So now that you have a perspective on the process, I'm going to dive into the review, show how we discussed these topics, how we integrated everything. And the first thing I want to say is that a paper like this, a review like this, you know, it's supposed to be read by a very large audience. For example, in this talk, for sure, we have multiple different backgrounds. I'm a bioengineer. I think it's a workshop and a focus on photonics. So for sure, we are all from very distinct fields. And writing a paper like this, you need to acknowledge that, and to know that maybe someone that is working on a completely different field is still interested in learning about it. And a Nature review should be accessible to anyone, you shouldn't have to be an expert on anything. So that's how we start the paper in mind. So we start framing our ideas and presenting it to an audience that might not be very familiar with the concept of tissue and mechanics and so on. And so the first perspective we give is a parallel between tissue engineering and materials engineering, okay? And here I'm going to be showing some different figures and making it exciting, but all of this discussion is in the paper. And we basically start by outlining that materials engineering is the design and tuning of the structure of materials to obtain a desired set of properties. This is done by engineers, by ourselves, okay? And tissues, they are basically engineered by nature, which designs and tunes cell matrices over times and evolution in order to obtain biological function. And we are trying, when we do tissue engineering, or when we try to do tissue engineering, basically try to do the same thing that nature does, but by ourselves. And of course, that's extremely complex. And to have a day where we can actually produce full organs and deliver them to patients, it's still going to take a long time, because organs have very complex shape and architecture and cell types, but they have also very complex mechanical properties. And mechanical properties themselves can be extremely important for the function of tissues and the function of organs. And this is the main perspective we want to show here. And this whole story begins around 2005 when Disher and all, they published this science paper that said that tissue cells could feel and respond to the stiffness of their surroundings. Okay, this was game changing because so far mostly people were focused on biochemical cues. It was not very clear. And from this publication onwards, we really realized that cells and stiffness can be a modulator of cells as important as biochemical cues, because cells, they are constantly moving around, grabbing, pulling, pushing their surroundings, sensing their mechanics, and it completely dictates the function of cells, therefore tissues and organs. So more and more we realize how important stiffness is. And what is stiffness, right? What is stiffness? We all know what is stiffness because it's an inherent maybe we don't even know the word when we're very little, but we already know that when we are trying to deform something, when if we pick a little piece of bread, it's something very soft, right? If we are trying to bend rubber, we see it's already stronger. And if we see steel, we know that we cannot move it. So we already know that bread is softer than steel. This is something that we know inherently. And we know it because we try to deform things with our hands, just like cells do to their environments. And we know that sometimes we need to make more force or less force for deformation. Sometimes we cannot really deform. So stiffness is a concept that we already have in ourselves. But to make an audience that can be very from general backgrounds to get into it, you need to show how stiffness translates into scientific concepts, into data that we can actually analyze and that we can actually explain. So this is how we start the review. And if you see it, we really compare these different materials. We really make these fundamental comparisons, so it's easier to fall and to draw into the rest of the document. So we start by saying that and showing the main relationships behind stiffness. Okay, so stiffness is just very simply the relationship between stress and deformation. So stress, when we apply a force, when we try to change the shape of a material, it will deform, and that deformation will, as you can see here, can be very different. It can be tensional, it can be compressive, shear, torsional. So there are different types of deformation that we can consider, and this was all very discussed in the paper. But what we wanted to show here is to tell you how we can extract this as data, right? So if you deform a material, you're going to get a curve like this, where to get strain increasing, so to deform the material gradually, you need to apply more and more stress. And basically the relationship or the slope of this linear curve is going to give you a modulus, an elastic modulus, a Young's modulus, which is basically the relationship between stress and strain. The higher the modulus, the higher the stress you need to apply, the stiffer the material. And modulus is usually used for materials. Stiffness is more structural, but they are connected with each other because if you have higher modulus, you have higher stiffness and so on. Okay, so here at this stage, even if you never heard of stiffness, you can already understand that it's a simple relationship between the force and the deformation. Okay, but still, if you never worked with this and you see that stiffness and modulus usually are given in Pascal, it's hard to have a notion of what is soft and what is stiff. If I say a material has stiffnesses or modulus of 10 kilopascal, maybe you think it's too much, maybe you think it's too little. It depends if you already understand and work with this. So if you look into the paper, and we wanted to find something that everyone could identify, a material that everyone could know and give a modulus for it so that they could all feel and have a perception of that, because that's how you can address a very big audience. And so we were thinking what materials we could use. Most materials like wood, they are very stiff. And then we had the idea of food, because everyone knows food, right? We all have to eat, we all have to touch it. So it's something that we all know. And that's how we give a concept of different ranges of stiffnesses. We start in the nature reviews by discussing food, all right? And here you can see, for example, different food, they have different modulus of stiffness, right? Panna cotta, which is sort of a soft, milky gelatin, has values modulus on around 100 to 1 kilopascal. Bananas and gummy bears, they are on the 50 to 100 kilopascal range. Apples are 1 megapascal, carrots are 7 megapascal, and so on. All right. And so with this, it's possible to already know, because everyone knows how stiff is a banana. Okay. So you can know that that's around 50 kilopascal. So if I tell you that fat is 10 kilopascal, you see that fat is like 5 times softer than a banana. And this is a very good way to give a perspective, because everyone is familiar with these concepts. You don't even have to be a scientist. And actually, an interesting point is that we only have bananas, apples, carrots in the beginning. But then one of the peer reviewers, he liked so much the idea of food that he said, I think the authors miss a softer food that can cover the range of 100, 1 kilopascal, which some tissues also have, as we will see ahead. And the peer reviewer said, may I suggest panna cotta? I had never eaten or tried panna cotta in my life. I didn't even know very well what it was. I had to go through a lot of papers to find some publications that actually assessed the stiffness of panna cotta, of this dessert. And I found some food papers that actually had it, and that it indeed had those mechanics, and it fit very well into the story. So just an example of little things and little details that were actually improved by reviewers, which liked an original idea and an original way to demonstrate things, okay? So now that we talked about food, of course, we can also make the parallel with living tissues. We all know that fat is softer than muscle. We all know that bone is stiffer, but how can we address this? How can we get the values from mechanical properties, from tissues, and from structures? And this is where we have to present a big bunch of data. So we have to show what are the techniques, what is the concept, what information do they give you, and that's where you should use a table. Okay, because a table allows you to condensate a lot of information. You have to write it down, you're gonna eat away all the words. So we focus on tables and figures to do that. And so basically, I'm not gonna detail it, but you can see that there are different techniques that you can apply. They have different concepts. They're gonna give different types of modulus, samples that you can use. Sometimes you can use living tissues, sometimes you can use fixed samples, so it depends on the technique and the dimension on which it informs, micro, macro, or nano. And that's also very important, because if you're looking at a tissue from a micro scale, the mechanics are going to be completely different than the bulk structure. If you look to a full bone, or if you go to a micro part of the bone, it's going to be very different. And a lot of times people make this comparison, which is not correct. And this is one of the topics we also discuss. You should always consider dimension, and you should only compare mechanical analysis if they are done at similar dimension. If you're going nano, you're just touching a collagen fiber. That has nothing to do with the full mechanics of the skin, for example. It's a specific part of it, but it's not representative. So that's something that we should consider. And so we took advantage of tables to bring along this information and to outline the different techniques for measuring stiffness. And once this was clear, so at this point, everyone knows what is stiffness. Everyone has an idea of modulus because we compared it with food. They can see how we measure it and extract this data in living tissues with the different techniques. So we can go through and start our discussion, namely, like the editors asked us, from the cell to organ level. And tissue mechanics, they basically always come down to cells and extracellular matrix, which are intimately related because cells make the matrix, but the matrix also influences the cells. So it's a very, very intricate puzzle that we actually had to dissect to try to create this gradual evolving perspective. And so we started with the smallest component, extracellular matrix. Once again, tables, they're very good for this because we can outline a lot of information, give values for things. So we started by showing some of the main extracellular matrices. So basically, if you zoom into your tissues, you're going to see cells residing into these fibrous networks, like you can see here in this figure of collagen. And this is the matrix. This is the three-dimensional structure that gives mechanical reinforcement to tissues, which is produced by cells, and you have it everywhere. And basically, you can see here, we brought together different types of ECM molecules and structures, techniques of analysis, the modulus. And now that you're already experts in fluid mechanics, you can see from this that ECM is quite stiff, megapascal, gigapascal. Okay, so far the stiffest thing we talked about was the apple, 1 megapascal. And you see that ECM actually goes to the gigapascal. And I'm gonna do a spoiler here and tell that this is the mechanics of bone. But the thing is, ECM is the stiff component, right? Which hints at cells being the soft component. But of course, we wanted to outline this perspective. Tables give a good information, and then we discussed some of the main structures. We discussed collagen, which of course is the most abundant ECM molecule we have in different tissues. There's 28 subtypes of it. It has very complex organization into fibrils, fibers, and networks. And then to show the particular mechanics of collagen and this concept of J-shaped stress strain curves, we write a little experiment that we can all do together right now. So if you have your hand or any part of your skin and you try to pinch and pull a little bit of it, it's very easy, right? It's a very soft, you can do this easily. But if you try to keep pulling, it's going to become extremely hard, extremely stiff and painful. And this is due to structures, and it happens also in other tissues. And of course, there's more things going on. But one of the reasons is collagen and its organization, because usually collagen is this kind of network. And when tissues begin to deform, collagen fibers start to align in the direction of that force. And then in the beginning, when you're pinching your skin, you're just moving around this random network. But then if you keep pulling it, the fibers are going to align and you're going to start to stretch collagen fibers itself. And like you remember from the previous slide, collagen is gigapascal thick, is extremely stiff. So when you start stretching it, it's very hard to do it. And that's why tissues become stiffer with stress. That's why they have these so-called J-shaped stress strain curves, because for initial strains, you need very little stress. But then as you want to move to higher strains, the stress really ramps up. And this is also a protection mechanism. It's why our skin doesn't fall apart, our organs, and so on. But collagen, of course, is very important. There are other molecules like elastin that gives elastic properties to organs and blood vessels, a very important ECM component for mechanics. The glycosaminoglycans, which are protein sugar molecules that you have, for example, in cartilage. And this was an idea also suggested by a reviewer. We didn't have the concept of pore elasticity set. The authors missed this concept. And I actually didn't know it at the time what it meant. So I had to go and do some research on it. And I had to do it a lot of times during this paper, go research topics that I had no idea they existed in order to be able to explain it to a broad audience. And basically, poroelasticity is a system that, for example, in our cartilage, these glycosaminoglycans, they are like a core of protein with sugars around. They can attract a lot of water. And basically, if you're stopped, your cartilage is going to be very stable. So you have these fibers and a lot of water around, but it's all stable. So you have a certain mechanic properties on it. And then if you start moving or exercising, like running, the compressive forces are basically going to pressurize the fluid around these molecules, the liquid around cells and ECM. And this is actually going to lead to an increase in the modulus, in the stiffness of the tissue. That's why cartilage is so important to protect it. As you exercise it, all the water given and brought by these molecules give it increasing in stiffness, so it becomes more resistant and it protects your bone. Okay, so poroelasticity, a very good, very interesting concept. We missed it the first time. Reviewers pointed it out, and we added it to the review. So this is the overview on different ECM that we looked on, and just to show you, because we also had this discussion, ECM is so important that very little deficiency in extracellular matrix molecules and proteins can have devastating health consequences. This is just the example of epidermolysis bullosa, which is a rare disease in which a single collagen molecule, especially important in the skin, is mutated, and there are different mutations and can have different outcomes. And basically, this resilience, mechanical resilience that we were demonstrating shortly before, it's lost. The layers of skin become unstable. They start detaching. A lot of blistering happens. And this is a very mild figure because I didn't want to impress you. There are much more severe cases of the disease. And it's a very hard to treat disease due to mechanical issues and collagen mutations. So just for you to have an idea of how important ECM is for our mechanics. And we will discuss also the topic of disease ahead. But before, remember, we are going from cells and ECM to organs. So we already talked about ECM. Now comes the concept of cells. And once again, there are hundreds of cell types. Okay, so how could we outline this? Once again, we took advantage of the table. And of course, here we didn't bring every single cell imaginable. We tried to have a representative view on it. Stem cells, bone cells, soft organ cells, fibroblasts, muscle cells, fat cells, cardiac cells. So we try to bring different types of cell types to create a complex landscape, of course, without going into all of them. And what you can see here, so all these modules are in the kilopascal. So you saw that ECM was megapascal, gigapascal. So clearly cells are the soft entity. And most of them you can see are on the 0.1 to 10 kilopascal range, possibly except keratinocytes. These are the cells that are on the top of our skin, kind of dry, kind of dead cells. So that's why it protects our skin, and they are stiffer cells or stiffer cell structures, but all others are very soft. Even bone, you see osteoblasts, 2.6, 6.5 kilopascal, they are soft cells, but bone is very stiff. That's due to extracellular matrix, okay? And how can we look into this? Probably you're curious if you've never seen this kind of work. How can we see mechanical properties of single cells? So we have to do deformations which are very small. And for that, we can take advantage of atomic force microscopy, for example, where a very small cantilever is going to do indentations on the cell, measure the deformation and the force it's making. And this relationship is going to give a stiffness. Nowadays, it's even possible to map stiffness in cells in different regions of living cells. So it's something that we are running. This is a paper from last year. So there's a lot of development in this area. And this was what we gave when the editor asked us, like, from cell to organ. This was the cell perspective. But then the peer reviewers looked into it, and I don't know exactly their background, and said, no. This is not enough. The authors are just showing the mechanics, but they have to discuss it. They have to discuss cellular cortex mechanics. They have to discuss the concept of tensegrity. They have to discuss cytoplasm and active gels. And this was something completely new. You know, I work at bigger dimensions. I never focused so much on the mechanicals of single cells. So this was actually the part that I had to learn more new things and go and explore and bring a lot of data and information to create a simple view on it. Okay. And so we started by showing and talking about the cortex, and I'm not going to go crazy here. I'm just going to give you a brief overview for you to have an idea of the different topics of integration. And basically the cortex, this is a structure that resides immediately below the cell membrane, a cross-linked mesh of polymers and biopolymers and filaments. And basically this cortex is like the protection of the whole cell, and it's what gives it a lot of its mechanical properties. We can also analyze it by doing tensions and compressions. It changes during different phases of the cell cycle when they are dividing, differentiation, and so on. And so we had to look and to explain the mechanical properties of the actomyosin cortex in a little bit a broad kind of view. And then look also at other interactions, not just about the cortex, but of the cytoskeleton. And one of them, and this was very interesting, was tensegrity. So tensegrity, I didn't know. I knew that it existed in architecture. It's like obtaining integrity through tension, but apparently it's also a mechanism that's very important to cells. And this was something that was established, the first papers by Donald Ingber, which is now the director of the Weiss Institute at Harvard, and the microfluidic chips and all those advances. And basically what his theory said in the beginning is that cells, they are always doing this tension to their environment, that's how also they become biomechanically stable. And I have a little video here to show you how cool tensegrity is. And you can see that the tension alone can actually hold things together. These are all soft springs. You can even then pile things up on it, and it's completely stable because these ropes are tensed upwards, this one downwards, and the structure is completely stable. It's amazing. And we have buildings that are actually built with this technology. Ballerinas and dancers, they use this to balance themselves when dancing together. But the thing is, it's also a very important part, and it also explains some of the cellular mechanical properties. But it's not all about tension inside a skeleton. There's also cytoplasm. So if you look into a cell, you'll see like all these yellow things here in this kid's representation. It's the cytoplasm. So cell is, there's a lot of liquid inside, right? We are made of water, cells are made of water. So there's this biggest component of the whole cell. There are the cytoskeleton structures, the organelles inside cytoplasm. But what is cytoplasm and what's the mechanics of it? So we also had to explore this topic as a suggestion from peer reviewers. And basic cytoplasm is a gel, is an hydrogel. What's an hydrogel? So maybe you're not familiar with it. An hydrogel basically is a water mesh, a cross-linked mesh of polymers. So you have hydrophilic polymers and you cross-link them and they form this mesh. And since they are hydrophilic, they attract a lot of water. And then basically you have a gel, which is like this one that we prepared here in the lab. This is a 99% water gel. So it's almost just water, but it's all stabilized by this mesh of polymeric structures. And if you look to the inside of the cell, it is something also like that. It's a little bit like a gel, but a particular gel, because cells, they also have the polymeric structures that draw in the water, but these structures are always polymerizing, depolymerizing. That's how cells can expand and contract and move around. So it's a gel, but it's active, right? Because it's constantly changing by the action of cell and the use of energy. So there's a whole concept behind it, which is the active gel physics and the active gel theory. Very interesting. I recommend you, if you want to go look into it, but we also review it in a general perspective in this paper, and we integrated that in order to understand the mechanics of cells from all perspectives. But like I said, all these different theories have to be applied, and we are still learning how to look to the mechanics of cells. So this paper, it's a Nature Materials publication from last month, where they challenge the concept that cell stiffness could depend on substrate mechanics. Okay, so even now, like 1 month ago, we are still learning new things about cells, and we had these different reviewers suggesting the different theories. So we thought, how can we discuss that they are all different and we have to bring them together and there's no unified theory? And for that, we use the concept of boxes. So these papers, they have boxes which allow you to have like a small discussion that is not main text, like a little review on the side that explains something. And we use this, and we actually discussed the fact that we don't have yet a single theory to explain cellular mechanics. We have to resort to different theories like tensegrity, active gels, cortex mechanics, in order to be able to understand the full picture. So this is an example of how you can take advantage of the review and the structure of the paper to have interesting discussions that are not part of the main text, but still very important. And I think this was actually something that we learn as we're going through all of these new topics. And it's very interesting, right? We don't know yet how to explain it. We have different theories altogether. It's like quantum mechanics and relativity. They don't yet fit. We still need a unified theory of everything, and the same applies for cells. Okay, so cells, we have seen ECM, we have seen cells, and then we built up to tissues. And we started by looking at cell-cell mechanics. So we all start as a single cell that divides and proliferates and becomes a multicellular structure, eventually a full organism. And there are also a lot of works that look to cell-cell mechanics, to these layers of cells, which can also be deformed, and you can also extract stiffness values from it. And not just layers, but also embryos. And you can see here, so these are embryos, and you see that they are still very soft, 0.1, 0.2, below 10 kilopascal, below 1 kilopascal even. So they are like single cells. So as cells proliferate and we have these multicellular structures, they are still very soft. So we wanted to go to reach tissues, but we are still at a very soft stage. So where does the stiffening happen? How do structures become gradually stiff? So we needed to find this link. And for that, we had to go and look into developmental biology. So the science that studies how tissues, organisms develop from birth. And so you can see here, not going to go through all of it, don't worry. But you see that as we divide, like as an embryo, as structures like an embryonic structure, the cells divide, there's a stage at which they start to deposit some extracellular matrix, some basement membrane, as you see here in these TEM figures. Okay? And so you see that cells are soft, multicellular structures are soft. But this basement membrane starts to be deposited. And here you see stiffnesses already going to the hundreds of kilopascals. So we already start to see the stiffening. And in fact, it keeps going from stage 6, 7, 8 stages of embryonic development. And here you see at stage 8 already values around 1000 kilopascals, so 1 megapascal. And if you degrade this matrix, it disappears entirely, the stiffening. So basically, stiffening begins when cells start to deposit these fibers and these matrices bind to it and start to form more stable and strong structures. So this was the missing link. This is the point where stiffening happens, and then we just develop and develop and develop. Some tissue and parts of the embryo go to bone, become very stiff, mineralize. Some parts still remain soft and give rise to soft organs. And after all of this perspective, we could get to actually the topic we had proposed first, the stiffness of living tissues. Okay, this was the first main idea. We want to be the first to review it all. We want to be the first to show a full figure that covers all living tissues and a range of mechanics of the organism. And this is it. And you see that it goes from very soft. We start with mucus, which is not really a tissue, but just for comparison. And then we have nervous system, so brain, and around 1 kilopascal. So if you remember, that's like the panna cotta, the very soft dessert. That's our brain. Then it's followed by all the abdominal organs and all the soft organs. And eventually we get to the stiffer part of it. So we spend an organism like ours from the Pascal to the gigapascal range. We covered it all. There's almost a tissue for all of these values. And we created this division. Until this paper, there wasn't a clear division on what we can consider soft and what we consider stiff. So we said, okay, then let's create this distinction. And we decided to create this distinction at 1 megapascal. So the stiffness of an apple. If you have something in the body softer than an apple, you call it soft. If it's stiffer, we can call it stiff. And stiffer tissues, you see, it's the interfaces in nerve and cartilage, stiff, ligaments, tendons, bones. Those are our stiff tissues. All others are likely on the soft region. And you see bone stiffnesses, they go up to the tens of gigapascal. And for comparison, titanium is around 100 gigapascal. So you see how strong our bone is. We have this full landscape. And then, of course, we show this figure, and it's just an overview because we had to look to hundreds of papers that look to different tissues from nano, micro, and macro mechanics. And we had all this data, and we created this figure, which is like the average bulk mechanics, but we didn't want to waste this data. So what we did is we picked all of it, and we created a set of supplementary tables that you can find online for free, where basically we put all that data going, all tissues, all ranges of mechanics from different animal models, humans, and also animals, different sizes of mechanicals, different types of modules. So create these very big supplementary tables that interested researchers, they can print it, they can go through them and quickly find, I want to see the micromechanics of a layer of skin. They just go there, skin, layer, micromechanics, and they can find the value. So sort of a little handbook of the mechanics of biological tissues. We couldn't put this anywhere, so we asked the editors, which is not very common for a review like this to have supplementary data. They accepted, and we gave these very big tables that contains all the data we investigated to create this discussion on the paper. And then we went through some topics that I'm not going to go into here, but you can find them on the document, like discussion on the dimension, scale, and direction of deformation, the anisotropy of the tissue, the fact that some tissues have orientation, like bone. If you do tensile or compressive, it's very strong because it's aligned like that. But if you do something that is like perpendicular force, it can break much more easily. Like that's why football players usually get fractures. The condition of tissues, fresh, fixed, makes a difference. The type of deformation and the nonlinear behavior, like we discussed for the skin and the collagen, the J-shaped kind of mechanics. So all of that has to be considered, and then that we discuss in the main text, and you can eventually look for it because it's an interesting thing to consider in order to compare tissues. And then, like I said, we also had to discuss health versus disease. So we need to find, our thought was, we have to find the disease cases where mechanics change. So we went through the literature, and there are like very different diseases where mechanics changing. So we said we cannot possibly have them all. We need to find the fundamental changes. We tried to bring it all together. We talked with doctors and physicians and asked, when does the mechanics of tissue changes? Because we wanted to simplify this to make it so that anyone can quickly understand. And we're basically to bring it down to two situations, which are almost nearly one: fibrotic and fibrosis events. So tissue stiffness increases in these cases. So when you have a wound, if you have a fibrotic liver, if you have a scar, because there's a lot of ECM deposition, a normal ECM deposition. Fibroblasts become myofibroblasts, which are contractile cells. They contract everything. They make it all stiffer. And so fibrosis is the main responsible of abnormal health stiffness in a variety of diseases. But there's a particular one, which is cancer, that not only has fibrosis, so ECM deposition and myofibroblast phenotype, but you also have these cancerogen cells that are going to be multiplying a lot. They're going growing fast, compressing the nearby tissue and causing an increase in what is named solid stress. Fibrosis and solid stress makes cancer very stiff in some cases. As you can see, in thyroid cancer, malignant tissue can have 10 times more stiffness than healthy tissue. And you can actually use stiffness as a method of diagnosis. And then we also discussed some chronic diseases like Alzheimer's, osteoporosis, which have also changes in mechanics. They're a little bit different, but it's always due to either changes in cells or ECM. Okay, and this was our main discussion on abnormal tissue states, something that the editors wanted us to include. And then in the end, the effort was to basically find the most fundamental basic concepts, which we brought down to these two: fibrosis and cancer. And then our final part, we are tissue engineering. So we are tissue engineers. How can we use all of this knowledge to build tissues? And once again, we wanted to bring it all together in a single figure that could be easy to understand. And we did it by this way. So we outlined static mechanics. This is like the classic approach: soft and stiff materials for soft, stiff tissues. But like hopefully you have seen in this presentation, we develop from soft to stiff. So it's not fair to expect a cell to immediately integrate a stiff environment, okay? Sometimes you need dynamicity. And dynamicity, lifelike changes can happen in time. So mechanics changing in time, which can be material intrinsic, like stress stiffening and stress relaxation, which are material properties. Or if the material is not so dynamic, maybe we can do it extrinsically, so we can ourselves do deformation and therefore allow cells to develop better by applying specific deformations. But in our organism, there's not just changes in time. For example, you might be aware that cartilage connects to bone. Bone connects to tendon. And in this connection, these interface tissues, there's a transition from bone to tendon, bone to cartilage, tendon to muscle. And this has a transition in mechanics, of course, accompanied by changes in cell type and so on. So we have not just changes in time, but also mechanical changes in space. And we also approach that from the point of view of stiffness gradients, mechanical gradients, and how to obtain them. Okay, so then we discussed all of this, and I'm just going to give you a brief overview for you to have an idea. Stress stiffening and stress relaxation dynamic properties of materials. Very, very interesting. These two Nature Material seminal papers from different groups, they came 30 November 2015, exactly in the same edition, and they showed different types of behavior that happen in biological tissues and are very important. Stress stiffening, basically when you have stresses, so forces applied over a certain critical value, these materials become stiffer. So the more force you apply, the stiffer the material becomes. And it exists, for example, in actin gels, collagen gels, fibrin, so a lot of biological structures that we have in the body. And it has been shown that taking advantage of this property can significantly affect stem cells, that the differentiation and the engineering of tissues. Stress relaxation, on the other hand, is kind of the opposite. So when you apply a stress, if you have something that is a covalent structure, the stress, you make a deformation, and the stress, you have to keep applying that same stress to keep that deformation. But materials like tissues, they actually have a viscous component. They're not just elastic. And what happens is that they gradually relax. So if you compress, for example, a collagen gel, you have to make some force for compression, but then you'll see that the material is going to cave in, the stress is going to decrease, and after a while, you don't have to make any force, the material assumes this new shape. So this is called stress relaxation. You apply stress, the material relaxes. And you can see it has a very significant effect on the behavior of cells, their differentiation, and it's also a dynamic property, which is very important to approach when you're trying to engineer a tissue. The other one, like I said, when materials cannot be so dynamic, we can ourselves do deformation. And sometimes it can be like tension and compression of structures. That's the most common things. But we can also have different types of deformation. And this is a very interesting paper, Nature Biomedical Engineering paper, where basically authors show that you could do nano vibration, right? Very, very small size vibration in engineered tissues, and that these vibrations could completely shift the response of cells and promote the formation of bone. Something very interesting. And there are different types of deformation, but the idea here is that we can also use forces to stimulate cells in a way that are going to direct them to form different tissues when you're trying to engineer them. And then, like I said, not only changes in time, but sometimes also changes in space are important. And this figure shows, this was all discussion in the paper, main text. I will try to go briefly over it, so we just had a section for it. But here I want to exemplify. So osteochondral tissue, that's cartilage to bone transition. You have changes in biochemical, biomechanical, electrical, structural, and metabolic. So everything is changing. But of course, here we were discussing mechanics. And this has been approached in tissue engineering by fabricating mechanical gradients. So you can make gradients with materials that you can actually have transition in the porosity of the structure, the mechanics of the structure, the ceramics of the structure. And this is actually a paper published here in our lab with Professor Rutkan Demirci, where they managed to create osteocondral gradients by taking advantage of temperature and an isotropy and orientation of materials. Just to show you that we engineers have the tools to make all of these things that happen in normal tissues. We have the tools to make them. Okay, so at this stage, it's important to wrap up the review. And in a paper like this, you want to set big questions for the future. And here you have some of them. And mostly we saw that these were like, how can soft structures reach the elastic moduli of stiffer structure, right? Can we have time-dependent changes in mechanics into stiffer materials? Because all of these time-dependent properties that I've shown you happen in a very soft environment. So can we also make it stiffer? Or somehow do we have to maturate tissues in a way that proliferation, deposition of extracellular matrix, and gradual step-by-step increases in mechanics might lead to stiffer entities? Like if we want to make bone, can we push stress stiffening to higher orders of magnitude? What are the approaches for it? How can we solve this in the future? How can we manipulate uniform materials to create changes in space, like gradients, like I've shown you? So we try to outline for us, looking through all the paper, main questions, and when we also added a section that discussed problems with mechanical analysis, and that's also part in our conclusion. So we see that there will never be a standard for analyzing biological tissues. They are very complex, very different. We always have to integrate results from different techniques, different dimensions, and try to translate it to materials. So we have to make this translation to materials in order to understand the complex mechanics of living tissues, which we are still learning, like I shown you, for example, in the perspective of cells. So it's important to show what is the gaps that still exist and to give directions to scientists that come after us and try to solve this in the future. And like I showed you now by the end, we actually have a lot of tools that allow us to have dynamic mechanics, mechanical stimulation, gradients of shape and mechanical properties. So we already have all these tools. We are understanding well and better the mechanics of tissues. So once we can understand how we can really synergize the soft material stimulation, stiffening of materials, and to reach the different mechanics of the very big spectrum that we have in our body, like I show you, probably by then, bringing all of this together, we could actually reach a final frontier and be able to engineer really lifelike tissues from the perspective of mechanics. Okay, because then there's shape, there's architecture, all of that, which is not the scope of this review. So now we're nearing the end, and a paper like this, you know, it goes to peer review. You might not have faced it yet. You might have already faced peer review. We sometimes hate it. I know. I also have problems with peer review before. You know, sometimes not pleasing, but when everything goes well, you want a paper that wraps up with this acknowledgement. The authors thank the peer reviewers for their constructive comments, suggestions that help shape the manuscript. Okay, this manuscript was really an effort from not just us, but a big editorial team, great editorial team from Nature Reviews Materials. Very good feedback from peer reviewers. They make it work hard, but they give very good suggestions, and that's what peer review should be about. So it should also be a lesson for all of us, which are we're writing papers and reviewing papers. We all should work towards a better manuscript, and here they really did. They really gave very good suggestions. Like I show you, a lot of the discussion came from peer reviewer suggestions. Actually, they were not our initial ideas. So by the end, the manuscript was much better, much bigger than our proposal, and we were very happy with the outcome. Okay. But now, acknowledging here the talk, so I, of course, have to thank everyone that I've been working with back in Portugal, the big research group, and especially the people that were involved with this paper. Here also now in California, I don't have a full group picture because of the coronavirus, so I couldn't get a picture with everyone. So instead, I got this picture just with a couple of colleagues, namely me and our host Rajiv. So this is a sunny day in San Francisco. And I want to just thank again for the invitation to be here, to have the opportunity to share our paper. Thank you all very much for your attention. And I leave you also with different ways that you can reach out to me. If you want a copy of the paper, I'll be happy to show you. If you want to discuss any topic, I'll also be able to do it. And I'm open for any questions you might have. Thank you very much.
Thank you, Carlos. It was very nice presentation, and I think you explained many things. And you remember, I started with there are many dirty work behind the skin. That
Yeah.
means each big paper actually it need a lot of effort, and also you need a lot of work to do. And I think finally you did this one and it's accepted, so we are very happy that you did a very good publication. And I would like to go directly to the question. So if you guys have any question, so I think it's actually a large number. You can see we have around 50 50 or 50 plus participant, and we have many professors from different part. Especially I can see yeah, we have our departmental chair, Mamun sir. uh, I would like to maybe ask Mamun sir, sir, are you like to ask any question or maybe any comment? Can you hear me, sir? Okay.
Okay, I can hear you.
Yeah. Please continue, sir.
Okay, thank you. Actually, I'm not good or knowledgeable in tissue engineering, but the thing is, these ideas actually, although these are the biological things, but there are so many things to do by the engineers. Okay, physical engineering subjects, man, they are doing these kinds of things. Really, it is amazing, and thanks to Carlos for actually showing this kind of research and describe these kind of things. And especially our young people, I think they have a good idea and opens a new avenue for many of them, I guess. So thank you very much, Carlos, for your nice presentation. Thank you.
Thank you.
Thank
Thank
you
you
very
very
much,
much
sir.
for the comments.
Thank you very much, sir, actually for your valuable time. And we can see also we have also other guests. I think so. We have Professor Mainul Hassan. So, Dr. Mainul Hassan, are you like to comment anything?
Thank you, Rajiv. It's a nice talk, Carlos. Very interesting. Although I don't work in this field, but it was really very entertaining to listen to you, and I learned a lot of new things. So, just had a question. I mean, when you write such big review papers, right? I mean, there's a lot of topics that you deal with, right? So, how do you stay focused on the theme, right? So.
Yeah, definitely. So that's an interesting point because it's hard to, of course, focus, especially if you're looking at very different topics. Probably you're not going to be expert in all of them, so you're going to have to integrate new topics, and then the question becomes, where should the focus be? What should I look for? So the strategy for that, it's really go through different perspectives, and you might also look into different types of reviews from previous years that focus on these smaller topics, and try to outline and identify which are the main contributions and the main steps in scientific development that actually led to where we are now, and try then to focus on those rather than doing a very broad and general discussion and trying to integrate every single paper possible. And I have to say that in some kinds of these discussions, which were probably more close to what I do and what I work, it was easier to understand because if you work in something, it's familiar to you. You know the contributions and you know the important steps. In some others, we actually had to take advantage of the suggestions we were getting. Some of these topics we were reviewing, and then we had these expert peer reviewers who were saying, this is not entirely correct. This theory is not very well accepted. Instead, it's more thought this and that. So we also took advantage of that and also worked with it in order to be able to identify these main works. And of course, the general focus of the paper can go very wide and very big. So my suggestion is always create very good outlines and structure very well the ideas before you start going into it. Otherwise, it's an ocean of literature, and we have more and more papers, and it's very hard to find a rational sense. So a very good outline, the topics you want to include, the discussions you want to include, and in a critical review, sometimes you are not going in such an integrative way that you look to all the papers and you try to discuss all of them. but you rather try to find key papers, key publications, and key steps that allow you to build this rational and go on from there. So you might build it in a little bit a different way than other reviews types, because you really need, like you said, to find the focus and to look to all of these perspectives and then try to derive the essential or the juiciest part of the fruit, so you can actually outline it and make it also simple for people that are not in the field. But yeah, it's an exercise, and for sure sometimes we got it wrong, but we actually were hopeful that we have this big team working on it, so we could always correct it and improve it. But that's a very good point. Sometimes it's hard to maintain focus in a review like that if you don't have a very good structure outline.
Thank you. Thank you so much.
Thank you for your question.
I think we have also like a many professor actually. So like a Kaji Maria, madam, can you make some comment for us? Can you hear me? Kaji Maria, madam.
Hello, Rajiv.
Thank you.
Can you hear me?
Yes, we can hear you. Please continue.
At first, I want to thank Carlos. It's a nice presentation. Actually, it's quite different from my field, but it's an easy presentation, and it's actually very informative, and I learn a lot. And thanks, Rajiv, to arrange this kind of webinar. Actually, it helps us a lot to learn many things.
Yes, thank you.
Thank you very
madam.
much for your comments.
Actually, I don't know like many people, so I would like to ask like so maybe you can unmute and ask the question. So we will be more happy if you ask more question. Please go ahead, unmute yourself. Just maybe you can give your introduction, like your background, or maybe from university you can give your affiliation, and then you can ask the question, please.
Hello.
Please go ahead.
Yeah, I am Mohammad Extracellular Rahman from Bangladesh, and now doing my PhD in Japan, in Japanese university. A question to Carlos. Thank you for your presentation. Carlos, for review writing, we basically need to summarize many papers, and we need to read many papers, and we have to organize these papers, and we need to find out main result from this paper. So basically, when we write their result, or we present the previous result, in this case, how about the plagiarism they define for this purpose?
Sorry, what do you mean, the region? I didn't get that part.
Yeah, plagiarism, how they want to check out the plagiarism, if because review basically we mentioned many papers and
Oh,
their
I see.
works.
So you mean like the similar sentences and words to other publications, right?
Yes.
Yeah. Well, so that's definitely important. So what happens is that is always checked, and more, specifically in these journals. And we actually had very different stages of the editors, and they wanted to confirm that everything we were writing was original, that all the figures were originally, because in this kind of paper, they don't want to have figures taken from other papers. They want to have original figures. So they wanted to confirm that everything was drawn by ourselves, everything was written by ourselves. For this particular paper, I'm not sure if it went through the software that some editors also use to detect similarities with other publications. We are not informed about that. But my advice to do it is you should write your paper, and that's what an exercise I did here, and I always do in my reviews. So first, I go through a lot of publications. I try to find the topic I want to discuss. I read the papers, I go through them. I write down the main ideas from these papers, but then I never use the same description that they use. I basically try to sum up the paper, the main idea that I want to discuss from that paper, and then I have all like my notes and my different ideas, and then I start writing by myself, not looking at all of the papers, just checking all of these notes and saying, so that from time to time we could see the evolution in this and that. There has been works that reported this property, there has been works that reported that. So you should write everything originally. That's the best strategy. I've also used some of the software sometimes to compare it with other publications, and I always have very little to no similarity to other papers. And that's because of this method that I usually use. So when you go through papers, extract your ideas, take your notes from them, but then try to write it without looking at the paper. So you just write it by yourself. And then, of course, you want to confirm if you're saying this is correct. Did they really say this in the paper? So you can go there and check it. But my advice is always to take your notes and then write it by yourself. If you write by yourself, then you can compare with the paper, you can make some changes, but you will see that you will never have any problem with plagiarism, no matter how many papers you go through. Just write it by yourself based on your notes, and by creating these 2 layers of differentiation from the paper, it will always be different, you know. And sometimes, of course, if you look to a very big paper, they will be able to find some parts that are similar, but you're gonna have like 1% or 2% similarities. That's not a problem. But if you do this, you take your notes and then you write it by yourself based on that, you will never face any problem of plagiarism. So that's my suggestion to you. It works very well for me also. I'm giving you that suggestion. Yeah.
Okay, thank you so much. Have a good day.
You're welcome. Good luck with your works.
Yeah. So if you have any other question, guys, just ask. Feel free to ask question. More question. I think it's more discussion. Actually, we want to increase our knowledge, so it's fine. Or even if you know, maybe if you have some confusion, you can ask.
Hello everyone. I would like to ask a question.
Go ahead.
Can you hear me?
Yep.
Okay, I am Samia Jaman from Buet. I'm a lecturer. So the question is, when you want to write review paper on others' papers basis, so how do you deal with the figures which contains ACM image or any kind of structures they have experimentally designed? So when I want to write the same thing on my review paper, how do I use those figures in my paper?
Okay, so that is a very good question, and that's going to depend on different types of papers. So some review papers you will see they demonstrate and they show the figures that have already been published. Okay, and this is something that you can do. That's something that is possible to do. You can go in and if you go to certain papers and if you want to reuse their figures, there's always going to be a part that is going to be about permissions, and you can actually ask the journal for a permission to republish that figure or to adapt that figure for your own publication. And in most cases, this is free and you can do it. Most journals, I have yet to find journals, some nature journals, sometimes they don't allow for this. They ask you to pay, but most journals, you can just go there, take the permission, and then you can use the figure freely. And that's one way to do it. So if you really want to show, and sometimes it happens, right? Sometimes you really want to show that SCM figure. Sometimes you really want to show that structure, and there's no way that a drawing will do. So that's the option. That's the option you can follow. The other way is if you want to show just ideas, or if you want to summarize, for example, in this case, if you have SEM figures and you want to summarize the structure, there are different words with different figures, and you want to summarize that sometimes you can have different mesh sizes, different types of structures, then you can think of a schematic drawing, which you can try to structure by yourself. And then you basically add all this information, you do the diagrams of changes in dimension and so on. And this is an original figure. Original figure are always good because you don't need permission. You can do it for yourself, and then other people will use it and will cite it. But sometimes we really have to use images, and even in nature reviews, they prefer original ones, but some reviews you find that they go to pick some papers images because sometimes you really need to show the figure. And when that is the case, you should check the journal, and you'll find that there's a place that says like copyright and permissions, and then they're going to give you like, I'm a student, I'm a faculty, I belong to university, I want to use figure 1 and I want to adapt it for publication in this publisher, and they are basically going to say, okay, you have been granted permission. So you can reuse figures, you can draw your own. It depends on how specific you need to be.
Okay, thank you, Carlos, for your reply. Another question. If I want to use data from their paper, so do I need permission from that journal?
Yeah, data is a little, Yeah, data
take
is a
their
little
data?
bit different, but usually I'm not sure. I've never reused a lot of data. I've read something about it. I think it's an interesting topic, and we are now going more to open data. So there are already papers that have data available, and in those it's very easy. You can just take it, pick it, and eventually cite the contribution, but you can use it. In other cases, I would definitely reach out to corresponding author or to the journal, depending on the publication, and be sure that it's okay to use. It should be, because we should all be able to use data. There's no point in repeating it. But sometimes authors do not provide it to editors. Sometimes it's not accessible. So that one, I think, is still a little bit under development in comparison to the use of figures. But for sure, there are already open data publications. And in the other ones, it's always, I mean, it's always easy to drop a mail to the editor or to the authors and ask for using data. If they are okay with that, they will give it to you for sure. If not, maybe they won't. But we are gradually evolving in this, you know, and more we see open data publications, more and more authors are pressured to give their data when required. So you should definitely, if you think you need something, you should definitely ask for it. Go ahead and ask for others if it's not openly available, just so that you don't have any copyright issues or anything like that. It's always safer to validate it. Yeah.
Okay, thank you. And lastly, it was a very nice talk.
Thank you.
Thank
Thank
you
you
for
very
your
much for your
question.
questions.
Yeah, please, if you guys have any more question, ask. Even
Hello,
if you have a
hi.
small question, yeah, go ahead, please.
Hi, Carlos. Very nice presentation, very interesting talk. It's Asif. I'm a researcher at British Telecom in the UK. So I have just a couple of questions. So what kind of software or platform you use to write your paper? And your quality of figures looks very interesting, actually. So
Yeah,
what do you use to make your figures? And the other one, like, do you suggest to publish or submit your paper into archive while your paper under review? Thanks.
Okay, very interesting points. So the first one, I write my papers in Microsoft Word. I don't think it's the best software, but you know, it always has to go to supervisors and to other people, and everyone just works with Word. So unfortunately, it's what we have to use, and I don't like it a lot of times. You know, you move a figure, it completely obliterates the document, so it's not very friendly sometimes. But it's mostly what I use due to having to interact with different people, and everyone knows how to use Word. I know there are also other very good software. I think LaTeX is amazing, especially if you are in the physics field and the equations field. It's very good. If you have supervisors that work with that, I'd go for it. And but in my case, it was Word. About the figures, that's also a very good point. So, like I said, this in these particular journals, you have the advantage that they have a team of designers, and no matter whatever you draw, they will redesign your figures. But I have to say that the figures I drew, they were only made minor changes, mostly in color, in some shapes, but they didn't change a lot. So I was very proud because it meant that I could make figures at that level. In my case, I use Inkscape software. So it's a free open source software. It doesn't have a very steep learning curve. It's relatively easy to learn. And it's very good to do simple 2D schematics, images, like the ones we have here in the publication. You can even sometimes give it a little bit of a 3D twist, depending on your drawing skills. But all of this, I like it a lot. It's a very simple software. So that's the one I use. But there are also many others. People also use Illustrated and all of that. I just use Inkscape because it's open access. It's free. It's very easy to use. So if you never tried, I recommend giving it a go. And for your last question about preprints, that's a topic that has been in my mind for a long time now recently, because we see more and more of these preprints. In the beginning, I wasn't sure what to think about it, and I have to say I never published a preprint yet. I still feel a little bit strange about sharing fully a paper that it's not yet published. But I think that that's just a matter of time. You know, it feels slightly uncomfortable because maybe you are not used to it. But I think that's definitely the future. You know, we are moving towards open access science. We are open towards open access publications, and I think the preprints are going to definitely be an important way to disseminate science. I think they also bring some problems because you basically have work that is going out in the world and is getting cited without being peer reviewed. And sometimes you find that this work has problems, but it has already been cited. People already built their research about it. So it's a very complex topic I don't want to go into. But if I recommend it or not, I haven't done it yet. I think it depends on the type of paper, but I feel that it's going to be more and more common in the future, and it's going to be a way to get our results out. And it's good sometimes, you know, you're working on something that you really want to push out there in the world before someone else comes and does it before you, because peer review has problems, right? I have a paper that is under review now, I don't know, for months. Okay, so in these months, maybe someone could come out and publish something like that, and it will already obliterate the impact of my own publication. So in that case, if I had a preprint out of it, I would already be protected. So I think that's a very good tool to explore for that purpose. And when you want to set your ground and set your position and go out with a paper, I would consider that. I would consider going with a preprint. So it's out. People know you did it, then you can publish it and review it. But it's a good way to get it out fast. You know, peer review sometimes is very frustratingly slow, and it takes forever for papers to go out.
Oh, great. I totally agree with your comments. Thanks.
Thank you very much for your questions.
Thank you, Asif. Thank you, Dr. Asif. Please go ahead. More question, please.
Hello.
Hi.
Hello. Carlos,
Hi.
I have a question. I'm Nazrul Islam, assistant professor at Marona Bhasan University. I have a question.
Sure. Go ahead.
What is the difference between survey paper and a review paper?
Well, there's a lot of differences, if I got it wrong. So you're asking between an experimental work, as an experimental paper, original paper, and the review paper, right? I guess that's your question. It's an entirely different document because, and it depends also on the kind of review. Some review is very oriented on research topics. So you'll find reviews that have mostly figures from other publications, and the goal is just to show like what has been done recently, what kind of results authors have obtained. And those kinds of reviews, they actually have figures that are very similar to research papers, to experimental works, and they are just bringing it all together into a single document. They are showing different figures of cells, ECM figures, and so on. And so those kind of papers are basically just outlining research. That's one type of review paper that maybe is closer to experimental works and basically just brings a lot of results together in a single publication. But then there's another level of reviews, which are such as like Nature reviews materials. And this kind of paper, you see, I show you here in my presentation, but all the paper figures, if you look into the publication, there's no published figure, there's no previously experimental result. It's all conceptual. Because in this kind of review, what you want to outline is mostly ideas and concepts and relationship between different things and bring together a lot of information in a document. And you want to do that, you cannot go and show specific cellular experiments, specific materials, experiments, specific paper figures. And that's why for this kind of paper, it's mostly focused on original content. They don't want to reproduce figures. They want you to draw your own figures because you're trying to bring together concepts more than actually specific results. And in that kind of review paper, it's very different from experimental work, and it's very different from other types of reviews, because what you want to outline are mostly ideas and knowledge that you want to bring to a broad audience, whereas a research paper is focused on a very specific audience that works on that field and is interested in your results. In this kind of review paper, you want to target students that are still doing their course, undergraduate. You want to give fundamentals to graduate students, but you also want to have new and recent results that interest faculty and professors, which are more prone to look to recent advances. So it's a very different scope. And the idea is behind concepts, the evolution of that specific science, and the main contributions over the years. It can be decades sometimes, it can be just the last recent years. So there's a lot of changes in the type of document, and in one like this, it's much more a conceptual paper, and you want to target a very broad audience. So that, I would say, is the main difference between a work like this and a research experimental publication.
Thank you,
Thank
Professor
you. Thank
Nazrul
you,
Islam.
Carlos.
Thank you. Thank you very much for the question.
Guys, if you have any more question, just go ahead, please. Unmute, just give yourself like your affiliation and ask the question. So, so if you don't have, maybe Carlos, I have some question.
Sure.
So for the review article, I think it's totally different. So you need like an invitation letter. And if you don't have like an invitation letter, that means like you need a very good cover letter to motivate the editor or the reviewer, mainly the editorial board. So can you share your experience, like how you write your cover letter? And also, like if you can also share your comment, like that you get from your reviewer, like what kind of question actually they ask and how you solve them. And so one question is like how you motivate them, the editorial board member, and then how you satisfy actually the reviewer, because yes, please go ahead.
So for the first question about the process itself, like I said, it starts if you are not invited, so you don't have an invitation, then the challenge is harder because you have to somehow convince them that the idea you have, it's worth of their attention and publication, right? And so that focus is on the proposal. And I briefly outline here what our proposal was about, but we indeed had to build an argument, you know, like we say, sometimes we have to make a little bit bigger of a drama than it actually is in order for our ideas to move forward. And here, basically, the main argument we had was, so look, there's all this research from the past 10, 20 years that is happening on the mechanical properties, effects on cells, and so on. There's this huge number of people. So this is another point, audience. You need to make them see that you have very broad audience. You know, they want the paper to be read a lot. They want the paper to be cited a lot because they need to maintain their high impact factor. So you need to explain why this appeals to a very broad audience. And so we went, you know, there are doctors that are interested in looking to the mechanics of the tissues and use it for diagnosis. We have tissue engineers, you know, they want to know, all of them that are working in skin and bone and cartilage, they want to look at different properties. We have biomaterials and material scientists. They want to know how can they design materials to be similar to biological tissues, right? We have people that are working in biophotonics. Okay, they want to know how can they integrate optical fibers into tissues, how can they have not just optical match but also mechanical match. You know, we have people from physics that are interested in the interaction between particles and tissues, which differ in their mechanics and also will affect their interaction. So we basically build this very compelling argument of why there is a big audience that's interested. And then we jump to the point where we basically say this has never been reviewed so far. And for a paper like this, it's important to have this kind of argument. And we said, so look, it's important for all of these people. It's important for all of these processes, but so far, no one has presented a review on this topic. We think it's important to have it. We think it is missing. And we think we need to target this audience that needs it on everyday science and work, okay? And then we said, so this is basically a perspective of materials from reviews. It's the first review of its kind. A lot of people need it. A lot of people are expecting it. So it should be published in a journal that has a broad reach, such as Nature Reviews Materials, okay? So this was our proposal. This is our concept. You build the idea, you tell why it's important. You have to show that there's a big audience of scientists interested in that topic and why. Ideally, it has never been reviewed so far, and therefore you bring it all together, a topic that is very important, interests to a lot of people, no one has reviewed so far, and then you have the secret ingredients for your cocktail to have it accepted, because that's what they want to hear. You know, they want new things that no one has done, that a lot of people are interested and are very important for science. You bring it all together in a document and you're gonna catch their attention. And that's how we caught their attention. Now, for the second part of your question about peer review, I actually didn't bring any of the comments because it was pages. It was very long. You know, we had comments that were as specific as saying the authors on reference 127 cite a paper that studied worms and not mammals. You know, they were this specific. They were checking the references we were using. They were seeing that maybe this paper uses a different animal model, so we should in the text say that we were using worms and not mammals. So they were this very specific, but they were also general. They also were like, in the beginning, like I said, we had this idea of going on to the mechanics. Then the editor said, okay, you should go from cell to tissue. And in cells, we just outlined the general mechanics of different cell types. And then we have 2 or 3 reviewers that said, I don't know, but I think they were from that field. Like, so for they focused there, and they said, okay, you are missing a lot of important works. You are missing the discussion of cellular cortex mechanics. This has been studied by Chera's lab, by Trepat lab, by this lab and that lab. You are missing the research on active gels. You should look into active gel physics series. You should look into this lab work, this lab work, this lab work. And it was like a lot of labs. It was not just their labs wanting citation from themselves. Okay? They were really giving us a full perspective. And then the challenge was, how can we integrate this, like you were saying? And basically, that's just effort that you really have to put into. So you have to really go read into these documents and then try to digest all this information to your audience. Because, you know, active gel physics, that Nature Physics paper, it's a very complex paper. It's full of physical equations, tensors, deformations, things that are very complex for someone that is, for example, working in biology. So we have to make a bridge, and that's the role of this Nature review. So you have to bridge complex science to a broad audience. So we basically had to pick the reviewers' comments, and we addressed every single one of them, okay? I don't think we disagreed on anything. We just said, okay, it's true, we missed that, so we developed here in this and this way. But then comes another problem, which is when reviewers have changing views and perspectives on a topic. So we had a reviewer that said, you are missing the concept of tensegrity, you have to discuss it, it's very important. We wrote it. And then on the second peer review, another reviewer said, this concept of tensegrity, it is not very well accepted. I don't believe in it. A lot of science don't think it's like that. So probably shouldn't discuss it. Okay. So we are at an impasse. We have one big reviewer that wants us to discuss it, one big reviewer that wants us not to discuss it. Okay. And that's something that you have to deal with in a political balanced kind of way. And in that case, what we did is what we discussed all perspectives. We created a box saying that we don't have yet a full picture and we have to use different theories. And we wrote in our response letter saying that, okay, we are not experts in these particular things. So we don't think we can say whether or not this field of science should be integrated or should be rejected. It has been published before. So we integrated it all and we think it should go like that. Editors agreed with us and we came out with this perspective, and we took advantage of this box system to actually outline these differences, the fact that not all people agree on the same theories, and that is still a very complex system. So to briefly sum it up, from the proposal point of view, you need to draw in big audience, have an innovative idea and something that has never been reviewed so far. From the peer review point of view, you have to try to really integrate everything. You're going to have very big suggestions. You're going to have to waste time. You know, these are really major reviews. The paper completely changes. You have to take the time, read a new topic, make it easy to understand, and then write about it. Okay. And in the end, the effort is worth it. But like you were saying, there's dirty work, there's hard work behind it for all of these points. And I hope I have created a detailed explanation for what you asked me.
Thank you, Carlos. You explained very well. Maybe I can request again the other people. Can you also ask more question if you like? Let me see. Maybe we have more question. So, Carlos, can you check also the message chat? I think there are some question more. Yeah.
Let me check. Okay, so we have feel free to write it if you have questions. I think we have a question here. How did you balance? Okay, so this is so the question here we have is like how to balance all the different papers in our homework, and then of course that there's variation on the topics, but how much time should we target to publish a review paper, and how many papers we should target to take preparation for this? So that's very general, and I think, like you say, it will depend a lot on topic and on type of paper and on type of journal. So for the first part, how to balance and our own work. So of course, some of these connected to my own work. I didn't show it here in the presentation because I really wanted to talk about the review. And I think that the biggest advances in this topic, they were not made by me, they were made by other people. But of course, I work also in this field. I work with the materials and cells, mechanical properties, high throughput platforms where we build gradients to condensate a lot of information to quickly detect the best stiffness value for different cell types and so on. So this is work that I've done and I have published. And my perspective is always good. And basically you combine your own experience to understand what's happening, what are the papers, what's important, what is good, what is not. But there's an important point, and this is outlined, and it's a rule of this kind of journals. You should not cite a lot of your own things. That's not the point. It's supposed to be a paper that goes through the whole picture. So if you're just focusing on studies from your lab or your own research, that's going to be a red flag for the editors. They're going to find it and they're going to say, look, you're talking a lot about your research, but what about the rest? So this is a topic. I mean, if you are really a new topic and most of the research is done by you, then for sure it makes sense to integrate all of your publications and to look more into those. But you should always have a broad perspective. I would always avoid just focusing on my own work because that's just selfish. And in most cases, for sure, there have been bigger contributions than your own, people that have done things in the past that you should consider. So of course you can also look into this. You should use your own experience, but you should limit to the point to do so. It should never make sense to focus a review paper just on your own work, because you should actually be fair to other people and demonstrate other people. And in this paper, we cited a lot of not so high impact factor publications, because there's also a trend that people always tend to cite big nature journals, big science journals. And of course, we also cited those papers. But sometimes, people don't publish there because they have a smaller work or they don't have the opportunity to go for such a big paper. But there's also good research in those papers. So if you see, and I think a lot of people were happy, they were cited in the Nature reviews, in like low impact factor publications and sometimes very small journals, because we scoured it. And if you have like good science, if they report a good result, even if there's just one figure or two figures, it's useful for the discussion. They should have an opportunity also to be cited here. So they were also integrated, and we also have a lot of these papers cited. And I was myself fighting for that. I was saying, no, I'm going to cite, if it's a small paper, if they have good research, if they have good science, I'm going to cite it. I'm not just going to cite the big guys, because sometimes small papers also deserve the attention. And in this review, we did it a lot. And that's important because we bring light to sometimes smaller groups that don't have the opportunity to publish so high. That doesn't mean that they do not do very good science. How much time we should target to publish a review paper? That depends on the dimension. This kind of paper, like I said, it took around 1 year. It was not so big, but there were a lot of topics. Like Rajiv was saying, we submitted recently an advanced materials paper, which basically took 5-6 months. So we took half of the time, and it's like almost 20,000 words, so way bigger, but it's a different type of review. And the process, of course, then we still have to go through peer review. That also takes time, but it depends. There's no time frame. You should feel free to do it as you prefer, go slower or faster. But one thing is important, with a review, if it takes too long, and I have had reviews that took like 3 or 4 years to develop, then you risk it becoming obsolete. If you have a review that is focused on recent work, you should publish it fast. Otherwise, it's going to become obsolete. And how many papers we should target? Well, that really depends on the size of the review. Sometimes you go for 200, 300 references. Some reviews only allow you to go up to 100. So the less references you have, the more selective you have to be. So usually it helps to scour a lot of papers and then to just highlight the more important ones and to focus on those as you go through and make your discussion. You're never going to be able to cover everything. So read the papers, see which ones you think are more interesting, fit by your discussion, and focus on those. Okay. So thank you for that question. And then
Carlos,
next,
we have more question. Yeah, can you check also? Yeah.
I'm gonna keep going.
Yeah, please go ahead, please.
So next question from Nishat is a lot of ideas. We need to get the audience. So this is a good question. How do we know if the idea will have a remarkable response? We don't know. Okay, we can really never predict, and this is for proposals, for research papers, and everything. We can only try. We try our best is always what we can do. So if you have different ideas, you should try to think Okay, so I have this idea. I'm working on this topic. Is it new? Is it not? Maybe not, but maybe I want to look into a different perspective. Who would be interesting? Who would be interested in reading this idea? And you would think, okay, maybe this can also be interesting to people working in healthcare. Maybe this can also be interesting to people working more in fundamental physics. I don't know. It depends on the topic, right? But you should try to think about how can we target a large audience. And sometimes maybe you can't. Maybe you want to do a very focused review, and that's also okay. But if you want to make something more broad, you should take time to think. How can I adapt this to different fields? If you're working, for example, in photonics, you could just talk about like light interaction, materials, and the way to develop it. But you can also think that there's always a biomedical perspective. Sometimes there's an healthcare perspective to it, and this is more my field, so of course I know it better. But it depends. It's not always easy, but if you want to do that exercise, just take the time to think and consider how can this be attractive to other people working in other fields, and then create discussions on that. Even if it's a new topic, it's not so well explored. If you have the idea, you try to go to the literature and you try to discuss that possibility. Like, how can we combine bioprinting with electron technology? I don't know. Maybe someone ever done it. But if you have an idea for it, and if you can pipe papers and connect these ideas, it's already enough, because it's a review. And the more novel you are and the newer perspectives you bring for the future, the better. So you can do these exercises. So let me go through questions. So there's another couple of questions.
So Carlos, he's actually Professor Atikur Ahad. He's
Okay.
a full professor, Tripoli Department, Dhaka University. Yeah. So yeah, go ahead, please.
Okay. So this reviews paper topic should be very focused domain, right? If it is broad and lack depth, analysis by the editor, and comment on this. It's, I mean, I would say it depends, and of course it depends on the editors and the journals and their interests. Of course, you will mostly focus in a domain or in a topic. Like in this case, we were interested in the mechanics of living tissues. But even though the topic can be focused, it should, and it's a balanced exercise. You need to have depth. You need to have scientific depth and interesting things that, of course, people are interested on. But it should also be broad enough that different backgrounds and people that work in distinct fields can still be drawn to the paper. So you have to balance both of those, and you have to understand that, of course, sometimes it's not easy, but it's doable. Okay, like I said, we were working on this paper. We were discussing very specific things. We were looking to the mechanics of cellular cortex. We were looking to the mechanics of cytoskeleton and tensegrity properties. We were looking to very specific physics behind it, but we were also discussing bananas and apples and carrots, right? So you can do both. And the idea is that if you want to draw a big audience, you need to make it so that you have an introduction that is broad and people can start familiarizing themselves in concepts. And then you go through different sections and you can focus on different topics, and you can go deep in those because you need to have depth. But that's an exercise you have to balance. But you can have both. You can have broad perspectives, but you can also have depth. And of course, that's a complex exercise. And as a bioengineer, people are always saying, you study all these different fields, so you know a little about everything, but a lot about nothing, right? So I hear this all the time. It's not true. You can have broad perspective and you can also have depth. You just need to have focus when you need to have it, but you also have to basically write things in a way that people can understand. If we're discussing physics with equations and very complex maths, you're going to be already limiting the audience. If instead you try to extrapolate those concepts and make them in a language that is easier to understand, even though still scientific, then you can already draw a bigger audience, and so on and so forth. But that's a very good point. And then another one, how do you deal with copyright issues? Publishers ask whether you have copyright or not. That's true. They will always ask you that. And if you're using figures from other papers, you need to have permission. If it's open access, it's very easy. You don't even have to ask for anything. You just need to cite the publication. If it's not, you need to use the legal processes and pathways that journals have to acquire permission. That's very important. In this paper, we used only original figures. So we draw them all by ourselves with the concepts that we extracted from multiple publications. So it was our original. We didn't need to ask for copyright, but still the editors asked us, like 3 or 4 times. If they were all original, and we had to sign a document saying that they were original. Okay, so it's all on you. If you're infringing copyrights, you're gonna have problems. So you should always get them when needed, or go for original figures and original images, and then you can avoid those copyrights. But yeah, that's also a very good point. So moving on to the next question.
Yeah, Carlos, I think we would like to thanks Professor Atikur Rahman Ahad for those nice questions. Thank you,
Yeah,
sir.
indeed. Thank you very much for very interesting points. And I see now you had that need to pay usually if not open access. It's true. Some journals you need to pay, but there are not so many actually. I'm collecting now some copyrights for some figures of a review we wrote recently, and most of them you actually don't need to pay under the concept of being a scholar on a university publishing from non-profit. But there are indeed some journals, namely, for example, nature journals, that might require you to pay a fee. And in that case, well, what I suggest is try to find another publication that might have a similar figure that you want to use, because it doesn't make sense, right? It doesn't make sense that we have to spend money just to highlight the work of others. Or if you can extract the information from that figures that you would have to pay for, but create an original schematics where you bring those ideas of different publications created by yourself, there's also a way to overcome copyright. So there are 2 different strategies to overcome that topic. But indeed, it's consideration, and it doesn't make sense, right? Why do we have to pay if we're just trying to highlight and give citations to other works in other journals? But sometimes we have to deal with that. So these would be my 2 suggestions: either try to find another publication, or alternatively try to create your own figure with a similar concepts, but a different one, so that you can also work around copyrights. But yeah, that's a very good point, Professor. Thank you for those questions.
Thank you very much, sir. Yeah, go ahead, Carlos. The other
Okay,
question.
so a lot of others. Another question is from Lubaba from Dhaka University. Maybe it's there are no naive questions. You know, I have naive questions all the time. I love to ask stupid questions because those are the ones that allow us to really understand things. So I wanted to know if I have any. Yeah, so this is very interesting actually, you know, and this is very interesting at this point, and I definitely think you should. If you find like there are trends going on and you're looking to different fields and you have a detection, that's perfect to go out with the review. Because, you know, when we're writing papers, a lot of times we want to find this kind of paper because we want to say that it has been shown or this trend has been found because we feel it, but no one has ever published on it. So sometimes these reviews on what is happening, how are the trends, or we are observing these trends and so on, we are seeing this, no one has ever reported it. That's a very good opportunity to have, even if it's a small and focused review paper, where you're showing that these type of properties change with this thing, or a review on the relationship or correlation between this and that. That's very good. Even if it's a very small review, they can draw a lot of citations because a lot of people is interested in this type of trends, in this type of evolution. So yeah, you can surely attempt to do that, and I would recommend you to do so. Especially if you are a master's student, that's going to be very good for you and to have a publication on the topic. So I definitely recommend that. And if you need help or suggestions for it, please get in touch. I'll be happy to give you some suggestions.
Carlos, I think I like to add something, because most of the students are undergrad or maybe the master student. So sometimes they think like, is it good to go for the review paper? So the thing is, like, if you work with a review paper, actually you will read many paper. So you will get actually so many idea. So if you read other people idea, so you can generate new idea. So that is the logic. So if you read more paper, that means more work, and then you can focus, okay, where is the gap, and then you can get a new topic so that you can work in future. So that is the goal of the review paper actually. Yeah.
And because, you know, if you're studying, you will be reading papers, and you're doing research, you'll be reading papers, but it's never such an exercise as writing a review, okay? Because sometimes you are just looking over papers quickly, you have an idea, but when you're writing a review, you really get a clear idea. So it's a very good exercise to do, especially if you are like on your master's, even undergraduate, you're interested in a topic. the review is the best exercise you can do to get familiar on a topic. It's the best thing. So every time I'm changing research, for example, when I came here to Stanford University, I started considering working a little bit with photonics. It was a completely new field. So my idea immediately was, let's try to do a review on the integration of all of that, because then I can do two things in once. I can learn, I can get ideas, and I publish a review paper. So it's a win-win situation, and you should never be afraid to do it. Go for it every time. It doesn't always have to be a big review.
Yeah, so I like to add, like that Carlos say, like when Carlos joined this group, so we start two together. So we start one review article and also like the research article. So currently the status is our review article is under review, advanced material, which is big journal. And we are also planning another journal, I think Nature Biotech, right, Carlos?
Yeah, something on those grounds.
Yeah, also like the same thing. So if you guys like want to work, is start parallelly. So you can start review article and also you can start the research article. So that means you can increase your knowledge, find the gap, and use this gap for your research. So that will be the goal, right? Yeah. Go ahead, Carlos.
Agree.
Please.
Okay, so continuing now, we have a question about evolution of research. That's actually a good point. And in different types of papers, I always like to present an evolution perspective on the topic, because I think it's always good to see how it evolved through time, but it is also hard to do. So the question here from Pooja is, do we have to look at the publishing year of the papers we have read in this case? That's one of the things, but it might be hard. So if it's a completely new topic, then you have to do it by yourself. And then you might really have to go through a lot of papers, find the oldest ones and try to find the first papers that appeared on the topic. And then you can try to build sort of an outline. It's dangerous to report like this is the first paper if you're not sure, because you can never go through all of them. And maybe there was someone that actually did it before. So finding the first is hard to do unless you can find previous reviews and a consensus within the literature that this paper was actually the first. When that happens, you can actually build up on that and you know that was the first, and then you can try to set up a timeline. But of course, it's hard to know which paper really exists, if there's something else or not. So we should try to avoid very specific saying this was the first, this was the second, this was the third, but rather we report an evolution. If we can find the first and then we just say, okay, afterwards there was this approach and these authors and these groups that followed this and this type of techniques to get this type and type of data. And from there you can build sort of a chronology. And then if you want to report specific works that you think are important, you can add a year to it. You know, sometimes people don't want to do it. I like to do it from time to time. I want to say, so you see, in 2010, this group reported this, this, and that. I'm not saying they are the first. I'm not saying they are the last. I'm saying this happened in 2010, because then I'm going to relate it to a paper that was published last year, and that helps you to have sort of a perspective. So if you want to do it, it's always good to try to find the beginning, the first papers that started work on that topic, because then from there you can build sort of a chronologic evolution, and you can have a bit of a timeline added to your paper, which I think is always an interesting perspective.
Yes, thank you, Pooja Roy. I think Carlos, so maybe you can also read the question. So can you go to the next question? Yeah.
So next one from Omira. I'm sorry if I pronounce some of these names wrong, but anyway, this question is, other authors and I have different writing styles. How can we overcome this issue? In case of publishing, does my background matter? So you're an undergrad student, so for now your background doesn't really matter. The background of your supervisors might matter, but not really yours. Okay, so it's good because you are free to explore anything. No one's gonna judge you on your background because you're still young and you're still starting and you're still exploring different fields. Although then, of course, the background of your institute, the background of your supervisor, that might have a weight sometimes. If you're from a completely different field, publishing something very different, editors might wonder if you are the best person for it. But you know, look at these Nature reviews, stiffness of living tissues. My supervisor in this paper, very big background in tissue engineering, so of course that makes sense. My own background, I have some papers on the topic, but there's a lot of it that was completely new, and it was not a problem. Okay, it's not a problem. If you want if you are giving scientific analysis to things and you're giving your effort to do a review, it doesn't really matter so much your background, as long as you do it thoroughly and supported by people that have actually worked and have experience in that. Now about the writing style, that's a very good point, you know, because I also have a different writing style and a lot of people, and I also had to deal with that issue, and I'm working sometimes in different groups and I have to connect that. And of course we all write differently, and the secret is that you should have someone who is the main responsible for the paper. In this case, if you are the first author, probably it's going to be on you, and you should have the target of uniformizing the language. So by the end, you want the paper to read, even if there are different contributors, you want it to read similarly. So you want to make sure that the English is similar and that the language is similar. And for the rest, it will depend on the people you are working with. Of course, for example, you write something and then your supervisor is probably going to correct it towards his style of writing, which sometimes is a little bit frustrating. My opinion is that we should always try to accept different styles of writing and just work on how to improve them, and not really try to change them to our own style, because we all have different styles. So if I'm seeing the paper of someone else, I should try to improve their paper and their style without trying to make them write in my own way, because we want diversity also in publications. Nevertheless, sometimes it's difficult, okay? And my suggestion to you as an undergrad student is take suggestions and writing and changes and corrections, take them always in a positive way. Look to them, try to learn from them, see what do you think is good, what do you think is bad, try to implement it in your own paper, try to move it forward, but don't lose your own style, you know, just try to keep your style, but improve it by all the feedback that you will have throughout your career, so that by the end you have your own style, but it has been trimmed and improved and it can be satisfactory to different people. So that's my suggestion. You know, I still deal with that problem a lot. Sometimes I really clash with other people because I write very differently. They don't like it. I don't like their writing. So we try to find common ground, try to learn from it, and then you'll see, okay, maybe I like to write in this way, but in science, it doesn't work. You know, I'm having a lot of problems writing in this way, so I'm going to learn and I'm going to change a little bit, but you should not just entirely accept changes, entirely accept corrections. You should also keep true to your writing style and basically try to find common ground. That's my suggestion. And that's a very good point.
Thanks a lot, Carlos.
Thank you. Thank you for the question. So next one from Lubaba. We have no precedence. That's why we can actually try to publish. Well, we can always try to publish, you know. And if we're talking about review papers, we don't have to have expertise in everything we are discussing. You know, we are basically underwriting a review. You have to see that you are doing a service to the community also in a way, because you're going to use your valuable time to scour to the literature and to make an overview of a topic that others can read and get into it. So you're actually giving a lot of you to the scientific community. So you don't always have to have a track record on that topic. You know, you are already putting the effort. If you're a scientist, you are learning how to do it, you're working on a new topic, you are doing a favor to the scientific community. So people are not going to be demanding that you have a specific background. You know, they want to see a review. It's on a topic they like, it's going to be helpful for them, and that's all that is. As long as it's well structured, scientifically sound, that's the main thing that you need for a review paper. Even if you want to go a little bit wild on a topic and explore something new, it's still fine. It's going to be peer reviewed. If there's something wrong, supervisor will tell you, peer reviewers will tell you. So all of that will come into the consideration of the final work. So never be afraid to explore a new topic and to go for a review. So next one. Okay, this is a big question. Let me read it quickly.
Carlos, sorry, this is not a question. Just this is my different, a little
Sure.
bit other
Go ahead.
comment. Yeah. So, yeah, just this is about like, I mean, regarding undergrad or master's students in my country, usually they get undergraduate students, they don't have free time to do research. So they basically do research within their courses in the final year. For master's students, some departments, they have 6 months only free only for research. And most of the cases, they don't have free time. I mean, within the 1 year or 1.5 year courses, they take all the courses, and in between they do research. So, I mean, basically they do not get enough time to study that much on a topic. So to do a research, I mean, definitely one shouldn't spend 30% or 40% time on reading only. And then if you start writing a review papers, then when will you start your own research and thinking and doing something? So that's my point, that I mean, if our students think that, okay, I should write a review paper because it looks easier than thinking and finding a solution and do a genuine research. So I think that this is a mistake, but considering Bangladeshi perspective. I mean, if you are a master's student for a few years or a PhD student for several years, then they can get enough time to learn on a topic and at the same time think and get into it, and then they can publish. This is my view, basically. So that is, I wrote, I mean, in a different point of view.
I see. Yeah. So let me go to that, because I agree with you, and my experience was exactly that. In Portugal, we also have a similar system. I had 6 months of pure research during my master's, where I had to produce research, produce my master's thesis, and do all of that. So I'm very well of that system, and that's also the system I grew up with. And what I'm saying is that it's also true. So if you have 6 months, you shouldn't spend half of it sitting by your desk writing a review paper. Okay, but you also can consider different types of possibilities. In my example, in my master's, I was also doing a 6-month master's. I was working on fundamental biology, and I knew that I was probably going to be contributing to a bigger work to come in years, and I would not be able to publish my own research paper because it was very fundamental. I was just going to have a part of it as my own contribution. And during my master's, this was not my suggestion, but actually at the time suggestion of my supervisor, a review came up, and the trick here was that we were all working on these different things. So we'd have to have our thesis. We needed to have an introduction. So we needed to perform a literature review anyway, because it's part of any master's thesis, right? So my supervisor thought, why don't we combine all of this, and you can have a part of a review focus on that. You can write part of the review focus on that, and basically wrote a collaborative review where it's not just focused on a single author like you will have maybe on a PhD program or a PhD that lasts 4 or 5 years. So we have much better time, like you said, to develop it. But rather we work a small part of the review, which was focused on our part of the work. Others wrote other parts, and in the end we published a very small cancer research review. It's already been cited like 50 times over the past 2 years, so it worked out very well. So there are ways also to overcome that. It's true, like you're saying, that if you have a master's student, if you're a master's student, you have 6 months, it's very hard to come up with a first paper full review written by yourself, but there might be room for a collaborative review, even with the supervisors or other people in the group, where you can just use this literature review part of the work that you're going to need anyway for your thesis, take part of it, write part of a review if you don't want to go for such a bigger paper. So that's, I agree with your opinion, and I think that it might be overcome in this way. You might break it down and have smaller contributions and have a review paper that is not just your own, but written by multiple people in the lab, for example. There are different ways to do it. Of course, you should not waste all of it for that. No way. Original work should always be a priority. I agree.
Yes, I think I agree also, Carlos. Actually, like a similar situation also happened. I know one guy, he was working with a master student during his masters, like the introduction section. He published I think it's a review paper with the impact factor 8. So even if you are a master student, I think if you can collaborate with other people, maybe like a 2 or 3 master students. So maybe each part from each master student, but the PI who can combine them together, so make it like a good review article. Or if you, even from your introduction, you can publish, even it's a small review paper, but you can do it if you have a patient to do it. Yeah. But yeah, actually,
of
this
course,
is,
there are very
yeah.
different formats. There are also mini reviews, which are really small in some journals, really focused, that you can write them up quickly also. Of course, you need to have support. If you're a student, you need to have support from your supervisor and other people in the lab that are going to be working together with you. But still, there are multiple ways that you can plan for and still manage to get a review publication of some sorts out of your own thesis and out of your master's, for example.
And also, I think there is another publishing system that you can go. It's like a prospective. So you can write, it's like a mini review. So you no need to write like the full review, but you can write maybe like a 6-7 pages, and maybe like a 3-4 figure, and you can go like a small review article. Most of the journal also they accept this. Yeah.
Agree.
I think maybe we have any other. I think we have more questions, Carlos.
Yeah, I think we had like
yeah,
some more
please
questions.
go
So,
ahead. Yeah, we want to make
next
all
one.
the people happy. Yeah.
Yeah, sure. I mean, I have the whole morning, so you can keep going. Anyway, reproducing simulation is a waste of time. I mean, I think it depends. So I've never worked a lot of simulations. Maybe Rajiv can actually help with this discussion. I would say that if it's something that you want to simulate, or it's something that is exactly similar to what has been published, you might be able to cite that simulation, and if you're you might be able to integrate it with your work without having to do it all over again. If there are differences, you probably will have to do the simulation from scratch. But I never done simulation, but surely
Yeah,
Rajiv
I
can
can go
also
ahead.
comment.
So yeah, so I think Rabiul Hassan, I think it's a good question actually. So if you try to reproduce the simulation result, I think it's good. It's not like a bad idea. So simulation is always support your experimental work. So like sometime we don't have the facilities. Like if you consider the country like Bangladesh, I know like most of the case we work for the modeling because we don't have like a many facilities like experimental work. So you can do the simulation, even the simulation work, they can publish even the nature.com, even the main nature they can publish. The thing is, if you do the solid simulation, that means a real concept that is really novel, you can go even higher journal, like over 10 imperfector you can go. And at the same time, like the reproduce, like if you think about the reproducing the simulation result, this is good. So you can reproduce the other people work, that is fine. But from that, you can use this expertise to make a new idea. That means you can make a new idea and try to model it. And if you can do this, I think that will be like a valuable. And at the same time, like if you go for experimental work, then with the simulation, actually you can predict the result, and then you can do the experimental work, and then you can side by side you can compare that, okay, this I got from the simulation, this is from experimental, so I think they are matching each other. So that's how actually simulation work. So simulation is to support your experimental work, but at the same time, if you don't have the facility for the experimental work, you can go only the simulation and go with the like a theoretical background, and even you can publish a good journal. Yeah, so you have a both option actually. Go ahead, Carlos.
Okay, so then I'm gonna jump on to the next question by Aminal. So what's considered a standard length for a review paper? And we might need a lot of instructions sometimes, it might be too lengthy. Advice to balance that. Yeah, that's a great point actually also. So about standard size, like Raji was just saying, it changes a lot. You know, you have mini reviews, which are like 2,000, 3,000 words. That's something like very like 2 or 3 full pages, or even less than that. You have reviews that require 20,000 words, which is like 50, 60 word pages. So they're very big. So it will always depend on the journal, on the type of review paper. Sometimes they have also like different types of review, like review, perspectives, progress reports. So it will always depend. And in a certain way, you need to have the idea of the publication that you want and the journal that you want to target in order to know what kind of paper to go for. Then eventually, if you have to resubmit it, you might have to make it a little bit bigger or a little bit smaller. But, and this is my opinion now, I think short reviews are much better, and they are actually the future. You know, nowadays there are so many papers coming out every day, you know, and people don't have the time. Who has the time to read 50 or 60 pages of reviews that are coming like 10 or 20 every day out? You know, no one has the time for that. So, and more, and this review is an example, it's a Nature reviews, and they said try to stay within 6,000 words. Okay, it's not a very big paper for such a long discussion and such a long topic, because the editors are also aware that if you go for a very big review, people are going to read the abstract and that's it. They're not going to go through the whole paper because they don't have time for it unless they are really, really interested. And then you are going to a smaller audience. So my advice is always to, of course, you need to consider the journal and the rules. If it's like advanced materials, they are going to always ask you for at least like 10,000 or 15,000 words. So there's no way you're going to publish something smaller in there because it's their policy. But if you don't have that regulation, you know, the smaller way you have to do, you manage to do it, and that's of course challenging because you need to integrate all those topics like you refer in a smaller document. So you're gonna have to be selective, focus on the main papers, on the main topics of discussion. But in the end, you know, it's gonna be a much more pleasant review. Everyone can read it faster, it can be approved faster, it can move faster. So I like small focus reviews. But like I say, depending on the journal, you might have to expand and go bigger. But my opinion is that if you can make something which is good, thorough, but small, that's perfect because that's what people are going to enjoy reading.
Maybe I missed previous question some part, like the how to validate simulation result. So if you done the modeling, so sometime you can also validate your result. Suppose if you do the modeling with respect to some experimental work, so you can model the result and you can publish. At the same time, you can say, look, this paper, like this reference paper, actually validate our simulation result. So like if we are at Bangladesh or other countries, what we can do, we can try to replicate from the big journal. Suppose from the Nature.com or Nature Photonics, you can go for the modeling. And if you can reproduce their result, that means experimental work, then you can say, look, I did this modeling, but the similar work, the experimentally, they also did this. So you can also refer their paper. That means actually you are validating yourself with reference paper. Okay, with this reference paper, my modeling is valid. So you can do like this, I think. Yeah, go ahead, Carlos. So if you guys have more question, you can ask. Yeah, if you don't have question, then we will end. But if you have more question, I think we are happy to go, because we would like to make happy everybody. So that if you have any small question, you can ask, and we are happy to go your question, please. So we will wait like 2 minutes, and then we will leave. So let me know.
Sure. And in the meantime, I'm leaving here all the different ways you can reach out to me. So if you have any questions afterwards, if you have any doubts, you want help to establish a review or something like that, feel free to drop me a mail. I'll be glad to help you. And me and Rajiv, we can also discuss if you have any topics that is more to his expertise or mine. So we can help you. Feel free to ask. And
if you have more questions, actually, you are happy to go. And I think Carlos,
go ahead.
Yeah, Carlos is going to publish also, like we have some like a big paper coming, we hope. And so if there are some good paper, I think we will come again. And also like the Carlos, I think we will request him to present again. I think so that like maybe there are many researchers, many students from Bangladesh, so that they can take this advantage.
Yeah, sure, because it's always a different perspective in their review and experimental paper, always very different things.
Yeah, so like this, like we work parallelly together, like the review article, also the research article. So we don't only focus the review article, so this review article to gather the knowledge, but it's not only your focus, because you need to go find the gap and go for the research. And at the same time, I think that the professor, like the Atikur Rahman Ahad, that mentioned, like if you have especially less, like a 6 month, like a master's project, so you need to be very focused. That means you have very less time. So you need to be balanced actually. Like you need to be balanced. If you want to write a review article, maybe like a short review, because you have a short time. At the same time, you need to find the gap, and also for your master's program. So you need to be very focused. So you have to do like this, And so if you like to ask any question, you can unmute yourself and you are free to ask the question actually. So actually we are reducing the number. So at the beginning it was like around 50, but I think we are going even less. So Carlos, maybe I think, so we would like to thank the Carlos. Actually, it's a long time we are discussing, and also it's very early at California. And I request him like that, okay, can you give me, because it's so early and this is also weekend. So most of the time we work in the lab. Even yesterday we are working 7-8 hours. And then in the weekend, I think maybe we have also planned for the experiment, but it's actually really hard because we have also like other work, research work. So I would like to thank the Carlos. Actually, he's very busy, with his experiment. I think 2 or 3 projects he's working, and it's really hard to get the time. So I would like Carlos to thanks from
My
Bangladeshi
pleasure.
people,
My
and
pleasure.
I think it will really help to the other people who are like who are young and doing research at Bangladesh. So, like we have very limitation. We have we have very less facilities, and we have like a limited resources. So actually, our people, they are trying to improve. Like in the research, we hope actually we will do like Europe in future, or Europe or North America or China maybe in future will be more stronger in research also. And so Carlos, we would like to thank you, and also like all the audience. And I think especially thanks like we actually get many professors from different universities, and we get like a many audience from like a different part of Bangladesh. I think we would like to thanks all of you, and we hope maybe in future we'll organize more event, and we will be happy. Please join, and please share your idea, and we will try to improve our country and make research environment much better. Okay, thank you, Carlos. So Carlos, would you like to make any comment?
Yeah, so I'll just like to wrap up by saying once again, thank you very much for the invitation. It's no effort at all. It's a pleasure and a privilege to be here and to share my perspective with all of you. I'm very glad for this session. I think it went very well. It was a very good start to this morning, to this Saturday morning, for you all, probably a wrap up of the day. And I just want to thank all the audience that were here showing some interest, everyone that asked questions, very good, very interesting points. And it's good to see that Bangladeshi science is alive and kicking, and I can't wait to look for the future collaborations and future advancements. I think that's going to be very exciting, and also future sessions here. I'm looking forward to all of that. And thank you very much. Thank you very much.
Yeah, thank you very much actually. And in future, I think if the Carlos, maybe we will plan also some research article, like a big research article we can present, and maybe we'll request you again. Maybe if you science advance, or maybe your advanced material paper come out, I think we can again request you. Okay. Sure. And thank you. I think it's a big difference, time difference. Again, we have to thank all the people. I think it's a big time difference also in Bangladesh, and some people, they are also from Japan. I think especially Atik Rahman Hassan. It's a very midnight.
1 a.m. 1 a.m. in Japan.
Yeah, it's
right?
midnight, and also like there are many people from different part of the world. So it's like we have a time difference. So thanks all for your valuable time and effort to join this meeting, and we hope we all together try to make our country much better. We'll share knowledge, we'll get knowledge, and we'll get benefit each other. Thank you very much. Thank you very much, all of you. Have a nice day, or have a nice night.
Thank you. Have a good night.
Good night. Bye-bye.
Bye-bye.
Bye.